Good afternoon, everyone. My name is Jewel, and I will be presenting today a part of my PhD work focusing on growth and substrate consumption of Pichia pastoris for the bioproduction of recombinant sweet proteins. Here is an overview of the different sections that will be covered in this presentation. First, I would like to give you a brief introduction on the topic itself and the relevance of the study. As you already know, bioprocess methods are widely used these days for the production of various commercially important products such as chemicals and enzymes. They have a wide application in food, chemical, and pharmaceutical industries. The ability of the microorganism to use simple organic substrates for growth permits the industrial scale cultivation of engineered cells for the production of recombinant proteins. Hence, an increasing number of chemicals are produced biochemically. To maintain this competitive position, continuous improvement of biochemical processes is essential. Great effort has been undertaken for improving yields, purities, and productivities of desired products through bioprocess engineering strategies since the very early beginning of the fermentation industry. Existing shortcomings of the microbial production system are addressed either by strain genetic engineering or at the level of the cultivation process in bioreactors. As an illustrative case study in this project, the bioproduction of sweet protein thaumatin is studied. Sugars play a major role in our daily diets. They are extensively used ingredients in numerous applications, especially in food and pharmaceuticals. Several studies point towards the fact that high intake of sugars can instigate risk factors such as high blood pressure and cardiovascular diseases. The food industry is exploiting the current situation by implementing calorie reduction into the consumables with a major focus on sweeteners or sugar replacers. Since the artificial sweeteners lose ground due to the potentially severe side effects imposed upon health, natural ingredients tend to be preferred in this novel sweetener markets. Sweeteners can be classified as caloric, low caloric, and non-caloric. The caloric sweeteners are also known as nutritive sweeteners. The non-nutritive sweeteners can be further classified into artificial and natural. The sweet proteins derived naturally from plants come into the limelight with the added advantage of not imposing any health issues, unlike the artificial sweeteners. The sweetest known substance on a single molecule level is a naturally occurring protein, thaumatin. It is natively produced from the katamfir fruit, which is grown in West Africa. Currently, the thaumatin used in food and beverage commercial settings is isolated from this fruit. Different forms of thaumatin have been identified, out of which thaumatin 1 and 2 are the most predominant forms. However, these plant-derived natural sweeteners exhibit limitations in commercial production in terms of raw material availability, taste quality, and cost-effective production. Hence, a biotechnological approach can be adopted to overcome these drawbacks through metabolic engineering of the cells. The methylotropic yeast Pichia pastoris has become a popular host for the expression of recombinant proteins due to the ability to produce foreign proteins at high levels using one of the strongest and most tightly regulated eukaryotic promoters, and its facility in performing many post-translational modifications. It also has the potential to produce very high biomass concentrations and large quantities of correctly processed recombinant proteins, both intracellularly or secreted. The extensive list of proteins that have been successfully produced with Pichia pastoris highlights the increasing future relevance of this microbial production system. Due to the emergence of Pichia pastoris to be utilized for the production of a substantial number of therapeutic proteins, it is of significant importance to study the factors influencing the growth and productivity of the organism throughout bioprocesses. Therefore, there is a need to optimize the process to achieve a high yield of the protein. Most of the bioprocesses involving Pichia pastoris are based on fed batch processes due to its advantage to maximize the cell densities and thereby resulting in maximum product yield. The standard protocol for the recombinant protein production by Pichia pastoris through AOX1 promoter is usually performed in 3 stages. First, cells are grown in batches on a defined medium containing glycerol to rapidly achieve high cell densities while repressing foreign gene expression since AOX1 promoter is repressed by unlimited growth in glycerol. Second, glycerol is fed at limiting concentrations in order to increase biomass concentration further. This phase allows the gradual de-repression of the enzymes necessary for the dissimilation of methanol and reduces the time necessary for the cells to adapt to grow on methanol. At the end of this phase, a sufficient amount of biomass is attained. In order to prevent the AOX repression by residual glycerol, a starvation phase can be introduced before the induction phase. Finally, recombinant protein production is induced by the addition of methanol in fed batch mode. Since the yeast is sensitive to methanol, the addition of the substrate to the medium must be controlled to maintain the concentration. The initial assessment of the growth of Pichia pastoris under different substrate concentrations using fed batch mode in a bioreactor can be a laborious task, which delivers results that are difficult to interpret. It is important to study the kinetics of microbial growth and substrate consumption in more detail under the same bioreactor conditions. Therefore, to understand the behavior of the organism with respect to the substrate concentration under relevant conditions, a series of growth experiments are conducted in a small-scale benchtop bioreactor in batch mode where the medium conditions are simpler. A schematic illustration of the correlations between living cell concentration, dissolved oxygen, and the limiting carbon source in a batch operation is shown here. In the next section of this presentation, I will briefly explain the experimental setup used for the batch experiments. The fermentation was carried out in a computer-controlled bioreactor with the possibility to control the pH, temperature, and dissolved oxygen. The conditions used in the batch experiments are as listed out in the table. The conditions for all the batch experiments were kept constant except for the substrate concentrations. Two strains of Pichia pastoris were selected, one being the wild type and the second a genetically modified organism capable of producing the protein. A wide range of concentrations were selected to study the effect of glycerol and methanol on the growth of Pichia pastoris. For the glycerol experiments, concentrations ranging from 5 grams per liter to 70 grams per liter was used. In case of methanol experiments, concentrations ranging from 5 grams per liter to 30 grams per liter was used. The substrate concentrations throughout the experiment was monitored using the gas chromatography and high-performance liquid chromatography. The glycerol samples were monitored using the HPLC, and the methanol samples were monitored using the GC. The method used for sample preparation was developed internally and are as described in the slides. In the following slides, I will be discussing the results attained from the batch experiments. The growth of genetically modified Pichia pastoris on different glycerol concentrations and consequential substrate consumption patterns for its growth and maintenance are represented here. The concentrations used were ranging from 5 grams per liter to 70 grams per liter. In this figure, the growth of wild-type Pichia pastoris on different glycerol concentrations and the substrate consumption can be seen. The concentrations used in this study were ranging from 10 grams per liter to 70 grams per liter. From both strains, we can see that the glycerol concentrations started to decrease extremely when the growth of Pichia pastoris entered the stationary phase. A batch glycerol concentration of 40 grams per liter is suggested in standard protocols, and according to published literature, higher concentrations of glycerol inhibits the growth of the organism. When comparing the kinetic parameters attained from the Barani and Roberts model, it can be noticed that both genetically modified and wild-type strain showcase a similar trend in growth when grown on glycerol. Upon increasing the concentration of glycerol in the medium to approximately 70 grams per liter, the growth rate was seen to reduce. However, a higher cell density was achieved at the highest concentration of glycerol. In this slide, a comparison on the growth rate of both genetically modified and wild-type Pichia pastoris grown in glycerol is shown. Growth kinetics of Pichia pastoris cultures are commonly described by models based on the Monod equation or inhibition growth models such as Haldane model. According to the results obtained from the batch experiments, the growth of Pichia pastoris on glycerol exhibits Haldane kinetics at a wide range of concentration. In the case of methanol batch experiments, the concentrations studied were ranging from 5 grams per liter to 30 grams per liter for the genetically modified Pichia pastoris. It was noticed that the growth of the organism was inhibited when a high concentration of methanol was used. A similar trend can also be noticed for the wild-type strain grown within the same concentrations of methanol. In both cases, when the initial methanol concentration was lower than 10 grams per liter, similar to the glycerol batch experiment, the methanol concentrations started to decrease extremely when the growth of Pichia pastoris entered the stationary phase. This pattern corresponded well with the published literature. Due to the toxicity of high concentrations of methanol on Pichia pastoris, higher cell densities are expected at lower concentrations of the substrate. But Pichia pastoris is still characterized by relatively low growth rates. From previous studies, it has been seen that growth inhibition occurs for Pichia pastoris in the presence of high levels of methanol. From the batch experiments, some discrepancies were noticed in the growth rate and cell density at high concentrations of methanol. On comparison of the growth rates of both strains on methanol, it can be observed that both strains exhibit a Haldane kinetics. In terms of the discrepancies noticed, repetitions are required to confirm the growth rates at lower methanol concentrations. Moving on to the conclusion and future perspectives. The aim of the study was to identify the influence of different substrate concentrations on the growth of Pichia pastoris, which plays a prominent role in the bioproduction industry today. Such studies, to understand the metabolic activities of the host organism, are relevant due to their contribution towards the improvement of yield and productivity of the recombinant product. The data collected from the substrate quantification during the batch process can be utilized for developing a macroscopic model aiming towards the production of thaumatin, sweet protein, and other recombinant proteins. This study, therefore, stands as a qualitative analysis for the prediction of extracellular protein concentrations as a function of available substrate concentrations. In the future, repetitions are required for the experiments with methanol due to the discrepancies noticed, and longer duration for the experiments are necessary to accurately estimate the growth parameters. Moreover, further optimization of the growth conditions has to be performed in bioreactors for more relevant conditions for aiming towards the production of thaumatin. These are the main published articles compared with the current study. Hereby, I conclude my presentation. Thank you very much for your attention.